You're a jellyfish, right?
No, that is so offensive. How dare you? I'm a Ctenophora. The jellyfish actually descended from me. Unlike that hyped up poser, I lack skin cells and I'm not in the phylum Cnidaria. Instead, I have 8 layers of cilia that contain these sticky organs called teens. They help me catch food.
So how'd you get to LMU then?
I usually feed on plankton in the spring and summer, but I was feeling festive, so I decided to come to LMU in the winter. I left my coastal, oceanic, and bottom-feeding friends to be here with you all today. Here is a picture of me from last summer. My size ranges from 1 millimeter to 1.5 meters. Ctenophoras in my taxa are diverse. I have a digestive tract with a mouth and 2 anal pores.
You left home to get here. Can you tell me a little bit more about that?
I primarily live off the coast, but I can also live in deep water too.
What other organisms do you live with?
Since I ate plankton, I live peacefully among Cnidaria, sponges, and echinodermatids. Ctenophores like me can have many different shapes. I can be bulbous shaped, like the Cydipid, have multiple lobes, like a lobular ctenophora, or have a pocket shaped, like a biroid.
I love your fit. What are you wearing today?
This is a picture of me in my juvenile larvae stage. I'm wearing 8 layers of mycelia that make me bioluminescent. They surround me on all sides, giving me radiosymmetry. Each taxa of Ctenophora usually contains a statocyte, an organ that controls the movement of the 8 comb rows. I usually have non-poisonous tentacles that help me catch food.
Do you want to get a bite to eat?
I usually eat plankton. I ate my kids for breakfast. Don't worry, my species are cannibals. Cannibalism is important for me because if there isn't enough food to eat, I'll just eat the larvae that I produce.
Do you have a significant other back home?
I am hermaphroditic. I carry both male and female parts. I can reproduce asexually or with a mate, but I only reproduce when there's a sufficient food source. Let me show you a video of myself swimming in water. To get around, I flap my cilia in and around my mouth. I also undulate the lobes on the sides of my body and sway my tentacles. The motion of my cilia creates a vacuum that pulls plankton toward my mouth.
I need to use the restroom. Do you need to go too?
Ah, yes, I could certainly excrete some waste from my 2 anal pores. Did you know that I'm a protozoan, which means my mouth filled up first and then my anus?
Okay, I'll meet you at the water fountain when we're done.
Great, I love water. I diffuse oxygen through my membranes because I lack a circulatory system, so this is how I breathe.
Tina, the Ctenophora, appears to be having a midlife crisis.
Are you okay, Tina? You seem to be having a midlife crisis. What's on your mind?
How dare you ask this on my mind? I don't even have a mind or a brain. I have nerve cords. My nerve cords create a nerve net.
Research also shows that the nerve cords in Ctenophora can help humans grow neurons. What's on your nerve cords?
I was just casually thinking about my evolutionary history. I've existed for 520 million years, since the early Cambrian.
You're probably thinking about your phylogenetic tree and your relationship to Porifera.
Research about my relationship to Porifera is contentious because my entire genome hasn't been isolated. I have muscles, nerves, and sticky teens that sponges don't have, and I don't know if I'm a common ancestor of a sponge or a descendant of a sponge.
This may clear some things up. This is a phylogenetic tree I created in my general biology lab at Loyola Marymount University. I used 16S rRNA sequencing to create it. According to it, you are a descendant of Porifera and closely related to Cnidaria.
I'm still unsure why I am shown as a descendant of Porifera. In a tree from a scholarly source, it shows me as one of the ancestors of Porifera.
And you aren't related to Cnidaria through sister taxa.
How can this be?
Hello, I'm a student in the Biology 111 class, and I used a character matrix to build a phylogenetic tree. You'll notice that my tree also shows Porifera as an ancestor of Ctenophora. Both of the trees we created in lab do not align with the tree we found from a scholarly source.
How do you reconcile the trees we made in lab with the trees we found from a scholarly source?
I would analyze these 3 trees through a weighted evidence approach. In the scholarly tree, I am an ancestor of Porifera. My genome wasn't fully sequenced. I contain similar genetic traits to Porifera.
Well, here's what I think. Because we created our matrices on an incomplete genome, we didn't consider how some traits may not have been shown in the phenotype. For example, sponges, a type of Porifera, have no embryonic tissues, while Ctenophora have 2. Ctenophora and Porifera both have genetic information that allows for the development of a nerve cord. However, in the genome of the Amphimedon queenslandica sponge, Porifera don't develop a nerve cord, though they have the necessary genetic information too. This could be why measuring physical characteristics based off of phenotype genres could be misleading.
Oh, look!
It's Dr. Willette's office.
We would like to thank Dr. Willett, Dr. Hertz, our TA Emily, and the LMU Biology Department.